Question number 106. Which of the following statements is incorrect about leg hemoglobin? First option, it is reddish or pink colored pigment. Second, it prevents oxygen poisoning during nitrogen fixation. Third, it induces root nodule formation. Fourth, it occurs in the cytosol of root nodule cells in legume plants. Which statement is incorrect about leg hemoglobin? Leg hemoglobin is a pink colored pigment which is found in leguminous plants. So this leg hemoglobin pigment is an oxygen scavenger. It scavenges oxygen, it traps oxygen and prevents the nitrogenase enzyme from oxygen as it is oxygen sensitive. So this leg hemoglobin is found in the cytosol of root nodule cells in leguminous plants. It is true. It prevents oxygen poisoning during nitrogen fixation. Yes. First, it is reddish or pink colored pigment. Yes, it is reddish or pink colored. Third one, it induces root nodule formation. No. Leg hemoglobin cannot induce root nodule formation during the nitrogen fixation. It is the node factors from the bacteria which induces root nodule formation. So the correct option for the given question is option number 3. Let's move on to the next question. Question number 107. Study the following columns and identify the mismatched option. In the column 1, you have given the bacteria. In column 2, this is mode of nutrition. And in the last column, it is the function of that bacteria. Say this first option. Rhizobium is heterotrophic in nature and it symbiotically fixes the nitrogen. It is true. Second, Anabaena is photoautotrophic. Yes, it is photosynthetic. It also symbiotically fixes the atmospheric nitrogen. Third one, Azotobacter, it is heterotrophic in nature, cannot synthesize its own food, and it performs free-living nitrogen fixation. So it is also correct. Fourth one, Nitrosomonas is a chemoautotrophic. Yes, it's a chemoautotrophic. It synthesizes its own food with the help of chemical energy, and it is a free-living nitrogen fixer. No. It does not help in nitrogen fixation. It cannot fix the atmospheric nitrogen because the fixation of atmospheric nitrogen is conversion of atmospheric nitrogen into ammonia, which cannot be done by Nitrosomonas. Nitrosomonas is a nitrifying bacteria which can convert this ammonia into nitrite. So this is the function of Nitrosomonas. So this option is mismatched 1. So the correct option for the given question is option number 4. Let's move on to the next question. Question number 108. For nitrogen fixation, which of the following is not a requirement of nitrogenase enzyme? First option, molybdenum. Second, high input of energy. Third, strong oxidizing agents for oxidation of atmospheric nitrogen into nitrate. Fourth one is anaerobic conditions. So, nitrogenase enzyme is required to fix atmospheric nitrogen into ammonia. So, those bacteria which contain nitrogenase enzyme are helpful to convert atmospheric nitrogen into ammonia. So, to fix atmospheric nitrogen, a nitrogenase enzyme requires molybdenum. Yes, because nitrogenase enzyme is a molybdenum iron protein, hence it requires molybdenum. High input of energy, yes, because without ATP consumption, the nitrogen fixation cannot take place. To produce 1 molecule of ammonia, we require 8 ATP molecules. So it is highly energy dependent process. Fourth, anaerobic conditions, yes, because nitrogenase enzyme is oxygen sensitive. It cannot tolerate oxygen. Hence, anaerobic conditions should be maintained, in which leg hemoglobin plays an important role. So third one, strong oxidizing agents for oxidation of nitrogen into nitrate. No. During biological nitrogen fixation, we require strong reducing agents which can reduce nitrogen into ammonia, not the oxidizing agents. And these reducing agents can be NADPH2 and FMNH2. So this statement is wrong. So the correct option for the given question is option number 3. Let's move on to the next question. Question number 109. Select the incorrectly matched pair. First option, primary CO2 fixation product in C3 cycle, PGA. Second, primary CO2 fixation product in C4 cycle, OAA, that is oxaloacetic acid. Third, primary CO2 acceptor in C4 plants, phosphoenol pyruvate, PEP. Fourth, primary CO2 acceptor in C3 plants is malic acid. Which one is incorrectly matched pair? Primary CO2 fixation in the C3 cycle or the Calvin cycle is a 3-carbon compound, that is PGA, phosphoglyceric acid. So this is correctly matched pair. Second, primary CO2 fixation is C4 cycle. C4 cycle is Hatch and Slack cycle in which the primary CO2 fixation product is a 4-carbon compound, that is oxaloacetic acid or OAA. So this is also correct. Third, primary CO2 acceptor in C4 plants. In C4 plants during CO2 fixation, the PEP or phosphoenol pyruvate accepts the carbon dioxide. It's a 3-carbon molecule. So this is also correct. Fourth, primary CO2 acceptor in C3 plants. During CO2 fixation, the primary CO2 acceptor in C3 plants is a 5-carbon compound, and that is RUBP. So this RUBP is a 5-carbon compound. It combines with the CO2 which has single carbon, and then it gives you 2 molecule of PGA, which is a 3-carbon compound. So this RUBP is the primary acceptor which accepts the CO2 and forms the PGA. So this is not the malic acid. So the correct option for the given question is option number 4. Let's move on to the next question. Question number 110. Non-cyclic photophosphorylation, first option, occurs in stroma lamellae only. Second, results in the formation of ATP, NADPH and evolution of O2. Third, involves 2 photosystems that work in series, first PS1 and then PS2. Fourth, is not associated with splitting of water. Non-cyclic photophosphorylation means ATP production or ATP synthesis during photosynthesis in non-cyclic manner, or the scheme called Z scheme, in which photosystem 1 and 2 both are involved. So it occurs in stroma lamellae only? No. Non-cyclic photophosphorylation occurs on thylakoid membrane. On stroma lamellae, there is a photophosphorylation called cyclic photophosphorylation. Occurs not the non-cyclic. So this option is ruled out. Second, it results in the formation of ATP, NADPH and evolution of O2. Yes, the splitting of water is also related with the PS2, and PS2 is involved in non-cyclic photophosphorylation. So evolution of oxygen and synthesis of ATP and NADPH is associated with the non-cyclic photophosphorylation, hence it is correct. Third, it involves 2 photosystems. Yes, it is true that it involves 2 photosystems that work in series. First PS1, no, it is first the PS2 and then the PS1. Hence this option is ruled out. Non-cyclic photophosphorylation is not associated with the splitting of water or photolysis of water. No, it is associated with the photolysis or splitting of water. Hence, there is evolution of O2 as it has oxygen evolving complex. So the correct option for the given question is option number 2. Let's move to the next question.